Peripheral membrane proteins are proteins that adhere only temporarily to the biological membrane with which they are associated. These molecules attach to integral membrane proteins or penetrate the peripheral regions of the lipid bilayer. They may interact with other proteins or directly with the lipid bilayer. An example of peripheral membrane protein is the cytochrome C in the electron transport chain. It is associated with the other integral membrane proteins of the electron transport chain, but it does not penetrate the inner mitochondrial membrane. Another example is phospholipase C. It's a class of enzymes that cleaves phospholipids and plays an important role in signal transduction. The phospholipid bilayer that forms the cell surface membrane consists of a hydrophobic inner core region sandwiched between two regions of hydrophilicity, one at the inner surface and one at the outer surface of the cell membrane. The association of a protein with a lipid bilayer may involve significant changes within the tertiary structure of a protein. The following are different types of interactions between the monotropic membrane protein and the membrane. First is the interaction by an amphipathic alpha helix parallel to the membrane plane. There is an in-plane membrane helix. The second one is an interaction by a hydrophobic loop. Third is the interaction by a covalently bound membrane lipid called lipidation. And finally, the electrostatic or ionic interactions with membrane lipids. Any positively charged protein will be attracted to a negatively charged membrane by nonspecific electrostatic interactions. It plays an important role in membrane targeting of electron carriers such as cytochrome C, and specific membrane targeting domains such as some pH domains, C1 domains, and C2 domains. Electrostatic interactions are strongly dependent on the ionic strength of the solution. One of the domains involved is FYVE zinc fingers. It is composed of two small beta hairpins, or also called zinc knuckles, followed by an alpha helix. It binds two zinc ions and has 8 potential zinc coordinating cysteine positions. Another one is the plextrin homology domain called pH domain, which is a protein domain of approximately 120 amino acids. It consists of 2 perpendicular anti-parallel beta sheets followed by a C-terminal amphipathic helix. It plays a role in recruiting proteins to different membranes, thus targeting them to appropriate cellular compartments or enabling them to interact with other components of the signal transduction pathway.